So, hi everyone, I am Abantika, and the topic that I'm going to discuss and briefly cover is going to be immunoinformatics and more. So, here in this workshop, which will be for a 4 days online workshop, we're going to cover reverse vaccinology, data analysis when it comes to a lot of immunological techniques which are used these days, and clinical case presentations. So, a brief background about me. So, I am currently a PhD student at Texas A&M University, and I am working on neuroimmunology. I have a master's degree from the University of Oxford, where my subject was also on immunology, but it was integrated immunology. So, I studied clinical immunology as well. Currently, one aspect of my work also involves identifying biomarkers associated with spinal cord injury using machine learning algorithms. And I'm very happy to connect with all of you on LinkedIn. So feel free to shoot me a message or connect with me. So what would be the workshop objectives? Briefly going through it and then discussing it in the following slides. So one is going to be a hands-on way of finding out which epitopes or which are the regions on the surface of an antigen that can trigger an immune response. So many vaccines these days are made in using reverse vaccinology and not the conventional vaccinology methodology. And that's what I'll be briefly talking to you about, and you'll understand what difference it has made in today's world. Then we'll also do some data analysis of flow cytometry, CRISPR-Cas9 gene editing based results on Western blots and ELISA. So these would all be from data which I have collected or people associated with me or like projects I've worked on. So we can do some data analysis based on those. And finally, we'll talk about some clinical case studies during the webinar that we'll be conducting for the 4 days. So whoever is new to the field, I'll just briefly introduce what the basis of vaccination was. So it began in around 1796 when the English doctor Edward Jenner noticed that milkmaids who had cowpox, so cowpox is this rare zoonotic infection. However, milkmaids who had cowpox were also protected from smallpox. Now, smallpox, as we know, is not there anymore, but it was a serious and often deadly viral infection. So Jenner had guessed that exposure to cowpox was protecting against smallpox, and there was a series of experiments done based on that. There were ethical concerns around it, but what logic was behind it is that when a person was exposed to the cowpox virus, their immune system recognizes specific viral proteins, which are these antigens. Now, these cowpox viruses, they share some antigens which are very common with the smallpox virus. And these shared antigens are similar enough that the antibodies produced against cowpox could also recognize and bind to smallpox antigens. And when later these individuals were exposed to these deadly virus, their immune system mounted a rapid and effective response due to the pre-existing antibodies. If you see this one here, this is where I would want you guys to pay attention to. In the past, cultivation of microorganisms, this is the conventional vaccinology way that we've all learned about, where causative organisms that needed to be cultivated in the lab, it had to be grown, and then individual components had to be dissected to see which one is the one which we can make a vaccine out of. Each fraction was assessed for efficacy. And then some antigen proteins, which are only in abundance, only they could be quantified. So, for example, you might have a really good protein which could be used, but it was not in abundance. So you cannot really go ahead with it. So it really took people decades to identify and grow targeted proteins. Whereas now, if we have reverse vaccinology, which are genome based approaches and pangenome approaches, as well as structural vaccinology, instead of the decades that it took, it has come down to like 1 to 2 years where you identify those proteins via a genomic based approach. So this reverse vaccinology was first time applied to develop a vaccine against serogroup B for meningitis. And this serogroup B for Neisseria meningitis was a major cause of sepsis and meningitis. And at the end of around 1990s, it was very apparent that a universal meningococcal vaccine was beyond the reach of a conventional vaccinology. So that's when the complete MEN-B genome was screened using bioinformatic algorithms, and then genome mining allowed prediction of approximately 600 novel vaccine candidates, 350 of which were tested for their ability to elicit a protective immunity. And in less than 18 months, reverse vaccinology enabled the identification of more vaccine candidates than had been discovered during the past 40 years by conventional methods. So this is one of the reasons why learning the basics of immunoinformatics and reverse vaccinology can be really helpful for you to understand what's going on in today's world and predicting immune responses to a new vaccine based on like protein sequence analysis, etc. So if you see this one, you can understand the different approaches that immunoinformatics has started taking over. So starting from disease prevention, disease treatment, disease diagnosis, prognosis, monitoring, drug discovery. So I'll try to cover two of them in the webinar that the workshop that we're going to have in the next month. So that's kind of the objective. And then moving on, how many databases would we learn? Multiple. So here I couldn't list out like all of them, but there are more than 20, 25 plus databases that we're going to work with. And one of them that we're going to work with is also going to be CEDAR, which is the Cancer Epitope Database and Analysis Resource. So you'll not really find a lot of videos or papers worked with CEDAR because it's usually a database or a resource which is cataloging antibody and T-cell peptidic epitopes in the context of cancer only. So we'll try to keep these workshop very interactive in a way that's approachable and understandable for most people, especially if you're a newcomer, you've never dealt with any of these databases, or you don't have a background in immunology, but you would really want to have an understanding. So this covers our reverse vaccinology and immunoinformatics approach. Now, if you see the immunotherapies, there are different immunotherapies that are in the market right now, and we'll walk through a lot of them, including CAR T-cell therapy and also the other alternatives like CAR-NK. And I can also discuss some of the advancements that are in the field right now. Apart from that, data analysis, I think this is very important because a lot of students I came across, even in the last workshop that was conducted, I think the team is going to share the testimonials from them very soon. So they really found this useful because when they want to do something in the field of immunology, they don't know how to analyze data, especially the ones which are important when it comes to flow cytometry. One uses flow cytometry very often these days. So how do you understand which immune cell population you're looking at? What populations are predominating in a certain disease conditions while it is absent in a healthy individual? How do you do this, which is called gating? So I will also be covering those aspects. And then more than that, the other interactive session we'll have is where all the participants can come together with me and we can have a real world clinical case study discussion where step by step you will act as if you are the physician or the attending clinician and you'll be able to look at reports and make your initial assumptions. So this is actually done to have an extended application of immunology. So like I mentioned, I have a degree which also involves clinical immunology. So I do have a set of real life clinical scenarios which we can discuss and see what is really happening in those cases, including the reports. And you can, after you know the basics, you yourself can be able to somehow come to conclusions as to what is happening. So I'll give you a very brief example for it here. This is just a brief one. It is much more detailed than that. So this is a case study where a small premature infant has come into the emergency room with issues with feeding, fever, and is also very drowsy. And then you did a lumbar puncture, and then it reveals that they have only 1200 WBCs per microliter. There's low glucose, elevated protein levels, and then you do a gram staining of the CSF and you see no organisms. And then there are other tests. And then based on all the tests and everything that you look at, you can deduce that it is meningitis, viral or bacterial. So if you go ahead and type in Google or PubMed, wherever, about immunoinformatics, you'll see the number of papers that have come up in today's, in recent times on immunoinformatic design and how different multivalent subunit vaccines are coming up against different pathogens using this approach, which is why I feel when you enroll in this course, it's going to be somewhere a very interactive and engaging discussion where you have a question, I'm here to answer. If there's something you have come up with, you have recently read, I'm also happy to learn about it and also give you insights about what I know about this particular aspect. So these were the things that I think would be important for us to cover in the 4 days workshop that is going to be there next month. And these would be, again, like I said, engaging sessions. In all my sessions previously, all the students have had really good time interacting with each other, interacting with me, and asking as many questions as they want. Other perks will definitely be discussed by the team, but I think the main perk is for me to share what I know, for you guys to know what is out there right now in the field. And we also have some take-home worksheets, which you can go ahead and do, and you can get back to me if you have any queries on those as well, and if you want any more take-home worksheets to run through. So I hope you guys had fun listening to the short presentation I had for you. And yeah, I hope you have a good one. Thank you.